Hi everyone, I am Clara Tang and I'm the Director of Research at KSF. And I'm excited to be giving you a research update today. And I will start by going over some highlights from our research program this year and then discuss some of the research grants that we have funded to accelerate specific treatments in our pipeline. So I'm going to focus on four of our research programs: Clinical Management Guidelines, KSOC, which is our Biomarker and Outcome Measure Consortium, our Research Toolkit, and Kabuki Count, which is our contact registry. To start us off, let's dive into the Clinical Management Guidelines. So one of our biggest research projects this year is the development of clinical management guidelines for Kabuki syndrome. These will give doctors everywhere a clear roadmap for how to care for people with Kabuki syndrome, even if they aren't specialists. So this work started more than 2 years ago and has grown into a global effort with experts and families working side by side. Last year, we brought together clinicians from around the world to review all the published medical research on Kabuki syndrome and draft recommendations. And we made sure that the community's voice was included at every step. Then this May, KSF hosted a consensus meeting where clinicians and 12 community members came together to combine medical expertise with lived experience. The guidelines will cover all major areas of care and include over 100 recommendations. We're now finalizing the manuscript with a goal of submitting it for publication next year. This will then be followed by a patient-friendly version that families can use. Next, I'd like to give you an update on how we're developing biomarkers and outcome measures through KSOC, our Kabuki Syndrome Biomarker and Outcome Measure Consortium. So, as Pat mentioned, we currently have 9 therapies in our pipeline. But for any of them to actually reach patients, we need to show that they're effective. And right now, there are no validated Kabuki syndrome specific biomarkers or outcome measures. And these tools are essential. They are what allow us to measure whether a treatment is effective, attract industry investment, unlock new funding, and ultimately move more therapies towards patients. And thanks to your support, in 2023, we raised 1.35 million to launch KSOC, a consortium dedicated to validating Kabuki syndrome specific biomarkers and outcome measures. The team is currently focused on 3 types of biomarkers: the DNA methylation signature, metabolomic markers, and event-related potentials measured with EEG. Many of you in the audience may have already taken part in KSOC by donating samples and sharing your data. And all of these measures will be compared with neurocognitive testing results to identify patterns that can serve as reliable, meaningful ways to measure treatment efficacy. And we are proud to announce that with your help, we have reached our goal of recruiting 150 families to participate in KSOC across 4 sites in the United States. The last few families are completing their appointments in October, and we expect to begin data analysis soon after. We are deeply grateful to everyone who has taken part in this project, and we could not have accomplished this so quickly and seamlessly without you, our dedicated and supportive community. Next, I would like to share more about our research toolkit we have been developing. This resource is designed to accelerate both current and future therapeutics in our pipeline by providing scientists with access to valuable tools to advance the study of Kabuki syndrome. So our virtual toolkit is housed on our website and marketed directly to researchers. You can explore some of these tools through the link that Kristen will put in the chat, or by scanning the QR code on your screen. And I'd like to highlight 4 of the tools we have been developing. So first, our cell line bank. We are establishing several Kabuki syndrome cell lines that will be stored and shared widely with researchers at little or no cost. These cells are derived directly from patients with Kabuki syndrome, making this project particularly exciting. And once available, labs can use them to study the molecular biology of Kabuki syndrome and test potential new therapeutics in a human-derived system. And later in the webinar, we will share more details about our fundraising efforts for the next step in this project. Next, our mouse models. We're working on establishing two Kabuki syndrome mouse lines that will be available for experiments and tissue collection. One line is already established at a contract research organization, CSCRO in the Netherlands, and a second colony is being set up at a lab in Chile. And I'll share how we're using them to accelerate the therapies in our pipeline a bit later. And we are also then creating and publishing protocols specific to Kabuki syndrome and our genes of interest, KMT2D and KDM6A. So, for example, we worked with a lab to test commercially available antibodies for KMT2D, determine the best one, and write up a detailed protocol for its use. This allows other labs to skip the trial and error process and immediately apply the best methods. And projects like this maximize efficiency, reduce redundancy, and accelerate progress towards treatments. Finally, many of you are already familiar with Kabuki Count, our contact registry. This is the largest contact registry for Kabuki syndrome worldwide. It not only tracks how many people have Kabuki syndrome, but also provides a way to reach them. This has enabled us to recruit for our own clinical studies, such as KSOC, and to support external researchers. In fact, it has allowed us to proactively approach experts in other fields and encourage them to study Kabuki syndrome by making participant recruitment seamless. For example, we recently recruited for a visual spatial study led by a vision specialist who had not previously studied Kabuki syndrome. But he took on Kabuki syndrome as one of his new areas of expertise because we were able to provide him with immediate access to interested participants. And as of last quarter, we had just under 1,300 people in the registry across 66 countries. And if you have not yet joined Kabuki Count, we invite you to do so now. Kristen will drop the link in the chat, or you can scan the QR code on your screen. We also need your help spreading the word. When you meet newly diagnosed families, please encourage them to join. Growing this registry is something that we can all do together to unite our community under one common goal. Those were just a snapshot of some of the research projects happening at KSF, and now I'll shift to give you an update on our therapeutic development pipeline, and starting with an overview of the therapeutic development process. So, to get an effective treatments to patients, we have to move through several steps. We need to learn more about Kabuki syndrome, like through our Discovery Grant program, build tools such as the biomarkers and outcome measures being developed in KSOC, test therapies and models like the cell lines and mouse models I described in our research toolkit, then test the most promising therapeutics in people through clinical trials. We're not quite at this stage yet, but we are laying the groundwork with projects like Kabuki Count so that when a therapy is ready, we can move quickly to recruit participants. Then finally, if the data show safety and efficacy, results are then submitted to regulators like the FDA, leading to hopefully approval and ultimately access to patients. So the process I just described supports all the therapies in our pipeline, which are the treatments we hope to move into clinical trials. Right now, we have 9 therapies in various stages of preclinical development. So on the left, you can see the list of potential treatments, and along the top, their stage in the development process. Green arrows represent progress made independently of KSF, while yellow arrows highlight progress funded by KSF. And as you can see, all of our therapies are still in the preclinical stage, but we are steadily moving forward. And today I will focus on the treatments marked with the yellow arrows to give you an update on the work that KSF has supported. So I'd like to start with our leading candidate, fepidamstat. So fepidamstat is a drug developed by the biotech company Horizon that targets a pathway that could potentially benefit people with Kabuki syndrome. To determine if this drug is effective and to help to risk future clinical trials in humans, KSF has funded a 300,000 preclinical study in mice. So the first step was establishing a Kabuki syndrome mouse colony at a contract research organization running the study. This took some time, but I'm happy to share that the colony is now established and breeding. And this was an important milestone because this colony becomes a resource that we can use for future research studies. As part of setting up this line, we needed to characterize the mice and identify a behavioral task where Kabuki mice show a clear deficit. Based on the crowdsourcing survey we shared, as Jessica shared earlier, we focused on task testing, learning, and memory. And after months of trialing different options, we think we've found the right task and are now working to verify those results. Since Fabidamstat is given orally, we also wanted to confirm that the drug actually reaches both the blood and the brain, and our results suggest that it does. And from here, we will look at biochemical markers like methylation signatures, RNA sequencing, and indicators of new brain cell growth to see if there are any cellular level changes. Then finally, we will assess whether the drug improves performance on the behavioral task. And with additional funding, the next step is to test Fepidamstat in human cell lines at Dr. Rosanna Wexford's lab at the University of Toronto. All these results will guide a go no-go decision on whether to begin clinical trial planning in humans. I hope to have the results of this study next year. Next, I would like to move on to another promising therapeutic that is in the therapeutic development stage, the modified Atkins diet and BHB or beta-hydroxybutyrate. As many of you know, several pilot studies have been conducted with people with Kabuki syndrome using the modified Atkins diet, which is a diet that is low in carbohydrates and high in fats and proteins. The evidence from these studies is promising, and the hypothetical mechanism of action is that these diets increase the levels of beta-hydroxybutyrate, BHB, in the body, and anecdotal evidence has shown that higher levels of BHB could influence cognitive improvement. Because the lifestyle changes required for these diets can be challenging, we wanted to investigate whether raising BHB levels through exogenous BHB supplements could be sufficient to produce therapeutic effects. The challenge with these supplements is that BHB levels spike briefly after each dose and then decline until the next dose. So whereas the modified Atkins diet produces more sustained BHB levels. So we need to determine whether these transient BHB spikes can have the same therapeutic effects as sustained levels. And so to test this, we started with mice. We're establishing a mouse model at a lab at Chile, which will be our second model ready for use in this experiment and future studies. Next, we're working on the dosing schedule, so determining both the dose and frequency required to raise BHB levels in the blood and brain. Once that is established, we will test whether these doses alter neuronal function or improve performance on behavioral tasks. And this work is aiming to start by the end of the year, and if we do identify a therapeutic effect of exogenous BHB, we will move forward with planning for a clinical trial as early as 2026. And next, I would like to discuss 3 of the candidates in our pipeline that all use a similar method, drug repurposing. So drug repurposing means taking drugs or therapeutics that are already on the market and using them for a new purpose. The advantage is that if these drugs are already proven safe, the path to patients can be much faster and less expensive. Our goal is to identify and develop existing FDA-approved drugs or nutraceuticals that have the potential to treat Kabuki syndrome. And we're pursuing this in 3 ways. So let's first talk about the project at Boston Children's Hospital, BCH. So we invested $110,000 in this effort to test candidate compounds that can boost either KMT2D or KDM6A in Kabuki syndrome human cell lines, and then move into our mouse model. The team has already found some promising candidates, and right now they're finishing the cell line experiments and beginning testing in mice. We also invested $100,000 in a collaborative project between Purposeful and Gen. This project uses AI to predict and prioritize candidate drugs that could improve Kabuki syndrome symptoms. And finally, we funded $66,500 in a project with Unravel Biosciences, which is focused on Kabuki syndrome type 2 caused by mutations in KDM6A. And the goal with this project is to identify potential repurposed drugs by analyzing patient samples. The collection of the samples is now complete, and so thank you to everyone who has donated your nasal samples, and we expect the report on candidate drugs within the next few weeks. Our next steps will be to continue testing the most promising candidates, especially those that identify across three repurposing approaches. By the end of these projects, we hope to add or progress several rows in our therapeutic pipeline. So far, the therapies we've talked about are drugs, nutraceuticals, or dietary changes. And now I'll briefly touch on our most recent discovery grant, which was awarded to David Genevieve at the University of Montpellier for his work on DCAS9, which is a gene targeting tool. So DCAS9 is a tool that can be used to increase gene expression. In Kabuki syndrome type 1, there is not enough functional KMT2D, so we're exploring the use of the DCAS9 system to restore KMT2D to functional levels. So first, the team will design a KMT2D specific DCAS9 system. They have evaluated the gene and believe they have developed a system that can effectively target our gene of interest. Next, they will test the system in zebrafish to assess its safety and to confirm that it successfully increased KMT2D expression. Ultimately, we want to know whether this tool can improve learning and memory. So the next step will be testing whether KMT2D upregulation enhances zebrafish performance on a learning and memory behavioral task. So it has been a very exciting year with new programs added and existing programs moving forward.